So let's continue with the properties of transition metals. We're going to look at hemoglobin, which is an example where we have a central metal ion surrounded by a multidentate ligand. So the multidentate ligand here is called a hem, which is going to form 4 coordinate bonds with your central metal ion, and another bond is going to come from a protein molecule that is called globin, and the remainder bond is formed between the central metal ion and oxygen or water. So when your blood moves into the lungs where there's a high concentration of oxygen, the water ligand there is replaced with the oxygen. So this is a perfect example of ligand exchange that takes place in our bodies. So there's something called so that's remember your oxygen and water ligands are interchangeable there. So there's something called carbon monoxide poisoning, where you breathe in carbon monoxide and it's going to attach and form a bond with the central metal ion there, but the bond that is formed in this case is stronger, and therefore it cannot be easily reversed. So your carbon monoxide will stick onto that site and prevent your oxygen from attaching, and this causes you to have oxygen starvation, and this may result in death. So that's called carbon monoxide poisoning. So remember, carbon monoxide is poisonous due to this process. Let's look at structural isomerism in these complex ions. So here we have an example of ion surrounded by this ligand that we looked at, this bidentate ligand that we looked at in previous slides, which is called ethane 1.2 diamine. So those are non-superimposable mirror images, and therefore we have optical isomerism in those complex ions. Okay. We also have cis-trans isomerism in complex ions. So in this case, we need 4 ligands of the same type and 2 ligands of a different type. This will cause cis-trans isomerism. So that's an example there. To my left there is the trans isomer, and to my right is the cis. You can see that the chlorine ligands there are at opposite sides and same side to my right. Okay, you can have cis-trans isomerism in square planar complexes, in which case you require 2 ligands of the same type and 2 ligands of a different type. So to my right there is the trans, to my left is the trans, and to my right is the cis isomer. So remember, cis-trans isomerism is also found in these complex ions. Let's look at how these complex ions form colors. This is important as you look at your qualitative analysis. When you put a transition metal ion into solution, it's going to form a colored solution. So that's one way you can know that this is a transition element. Non-transition elements will not form colored solutions. So what causes this? There's something called d orbital splitting. So when a ligand associates with this central metal ion, it's going to split the d orbitals into a lower energy orbital and a higher energy orbital. So there's going to be a gap when you split these orbitals, and the split is due to the ligand associating with your central metal ion. So there's an energy gap that is created. So when light is passed through this solution, electrons can be excited from a lower energy level there that has been created to a higher energy level that has been created. So electrons can be excited from a ground state to an excited state, and the amount of energy that they require to move from the ground state to a higher energy state is proportional to the frequency of light that they absorb. So they get this energy from the light that they absorb, and so the remainder frequencies will combine to give you the color. So let's remember, the size of the gap there depends on the type of central metal ion and the type of ligand. So we are saying here the light that is that you see is the remainder of the light that we passed through minus the frequencies that have been absorbed. For example, in copper there, the complex ion has copper surrounded by 6 ligands. So here, this complex ion is going to absorb frequencies in the red range, and the remainder frequencies will combine to give you that color, which is light blue. So the light blue is the remainder of the frequencies after the frequencies that match the gap have been absorbed. Okay, so let's remember that this is what happens. Okay, let's look at how the ligand affects this gap and the color that you see. So let's look at an example here where you have your cobalt surrounded by 6 water molecules, and you're going to exchange those 6 water ligands with 6 ammonia ligands. So this is ligand exchange. So what happens here is that you're going to see a color change. Why? Because the gap that is created there in the ground state and excited state when your orbitals split is dependent on the type of ligand. So here we see a pink color when cobalt is dissolved in water, but we start to see a straw color when we change the type of ligand. Okay, let's look at an example where you also change the type of ligand and you also change the coordination number. So here we are going to replace our 6 water ligands in that metal complex that involves copper and water, and we see with that when the copper associates with water there, we have a pale blue color. But when we replace with a larger ligand, chlorine in this case, your chloride ions, you're going to change the coordination number, you're going to change also the shape of the molecule, and you start to see a different color. So with chlorine, it's going to form a yellow color. Okay, let's look at another scenario. In this case, it's the oxidation state of the central metal ion that changes from 2+ to 3+. So the oxidation state also has a bearing on the color. So your ion is vanadium, where we have here our vanadium changing its oxidation state from 2+ to 3+. The ligand does not change, it's just the oxidation number, and this changes our color from violet to green. So this is all I have for now. So we'll continue with this series. So please share this video and like. You can also leave a question, leave a comment. See you in the next video.